In our last lecture, we talked about some of the structural changes that happen in the brain as we age. Our prefrontal cortex shrinks, our medial temporal lobes tend to deteriorate, and the white matter connections between brain areas often become less efficient. Well, as you might imagine, those structural changes affect how the brain functions, and that's what I want to talk about in this lecture. Now, it's natural to think that the kind of structural deterioration that we talked about last time would always be associated with a corresponding deterioration in neural function. And it certainly often is, and we'll talk about some of the ways that the brain's function declines with age in the first half of the lecture. But I'm delighted to report that there's also some good news. You see, our brains don't take their structural deterioration lying down. In fact, there's now substantial evidence that our brains can reorganize themselves, and that reorganization can sometimes compensate for structural deterioration, allowing us to perform significantly better than we otherwise would. So there's some good news in addition to the bad news, and we'll explore that good news in depth in the second half of the lecture. Now, if we want to investigate how age changes neural function, then we obviously need to study the brain in action while it's actually performing cognitive functions. But how can we do that? And specifically, how can we do that in aging human beings? After all, we can't just look inside someone's brain and watch neural activity unfold, can we? Well, actually, yes, we can to some extent. You see, back in the late 1980s and early 90s, some new techniques were developed that allow scientists to estimate neural activity in human beings without cracking open their skull. One of these techniques is called functional magnetic resonance imaging, or just fMRI for short. And since most of the work I'll be discussing today used fMRI, let me spend a few minutes talking about how it works. Now, as you may know, an MRI scanner looks like a big tube that you can lie down in. Well, that tube contains an extremely strong magnet. In fact, the magnetic field inside an MRI scanner is about 3 orders of magnitude stronger than a typical refrigerator magnet. That's strong enough to pull a pen out of your pocket, a wrench out of your hand, or earrings out of your ears. In fact, floor polishers, oxygen tanks, even hospital gurneys have been sucked into MRI scanners when they were accidentally brought too close. And that's why you can't have any iron-containing material in or on your body when you get an MRI scan. But otherwise, it's perfectly safe. Most dental fillings are also not a problem. Now, MRI scanners exploit their strong magnetic field in order to create images of the inside of your body. You see, you have water molecules throughout your body, and the protons in the hydrogen atoms of water molecules, they act like tiny little magnets. In particular, when you get inside an MRI scanner, those hydrogen protons inside the water molecules line up with the magnetic field. The scanner can then use radio frequency pulses to kick those protons out of alignment and into a higher energy state temporarily. However, after the pulse is over, the protons will relax back into alignment because that's a lower energy state. And when they do, they give off energy, and that can be detected by the scanner. And it turns out that how long it takes the protons to relax depends on the magnetic environment that the proton is in. Bone has different magnetic properties than muscle, and muscle has different magnetic properties than skin, and so on. And so the relaxation times of protons in these different tissues are different. And that means that if you measure relaxation times all over the body, you can actually create a 3-dimensional image of the body's interior, an image that distinguishes the bones, muscles, and other tissues from each other. Even tumors and other abnormalities have different magnetic properties, and that's why MRI is such a useful diagnostic tool. But how could MRI help you get images of the brain at work? In particular, is there any way to use it to create images of brain activity? Well, as a matter of fact, there is. You see, blood that's carrying oxygen also happens to have different magnetic properties than blood that's not carrying oxygen. And so with a few tweaks, MRI can be used to create images of where the oxygenated blood is located in the blood vessels of the brain. And because oxygenated blood gets sent to brain areas that are active, this technique can be used to create images of brain activity. And furthermore, by taking snapshots of oxygen levels every couple of seconds and then stringing those snapshots together, you can actually estimate changes in brain activity over time. That's functional MRI. And it has absolutely revolutionized the study of human brain function. And most relevant to us, it has revolutionized the study of age-related changes in brain function. So, what do we find when we use fMRI to estimate neural activity in younger and older people? A lot, actually. Now, I'll start by talking about some age-related changes that seem to reflect decline in neural function, but then I'll talk about changes that may actually reflect compensation, enabling us to perform better than we otherwise would have. Well, first, let's talk about declines in neural specificity. This topic is actually a major focus of my own research, and so the story here will be a little more personal. I ran my first fMRI study around 1995 when I was a postdoctoral fellow working with Martha Farah at the University of Pennsylvania. Now, at the time, I was interested in the neural basis of reading, and so I asked people to read letters and numbers while they were in the fMRI scanner. And then I examined the neural activity that was evoked. And I found something kind of surprising. People use very different parts of their brain to read letters and to read numbers. In particular, an area near the back and on the bottom of the left hemisphere seemed to respond selectively when people were processing letters and words, but not numbers. And a number of other scientists reported evidence for similar kinds of selectivity in neural response. For example, Nancy Kanwisher and her colleagues at the Massachusetts Institute of Technology found that nearby brain areas responded selectively to faces, while another area responded selectively to outdoor scenes like pictures of buildings. Yet another area responded selectively to body parts. Now, together, these studies demonstrated a fair degree of neural specificity in the brain's visual system, with different categories of stimuli being processed in different brain regions. But all these studies were done in young adults. So a natural question is whether that kind of neural specificity changes as we get older. I mean, one possibility is that as we gain more and more experience, maybe our neural representations become more and more specific. Or maybe neural specificity declines as we age, reflecting structural deterioration in the brain. Yet another possibility is that neural specificity doesn't change much after we reach adulthood. So which is it? Well, the desire to answer that question is what first drew me into aging research around the year 2000. One of my colleagues at the University of Michigan at the time was Dr. Denise Park, the aging specialist whose work we discussed at length in Lecture 1. We began collaborating, and we've maintained a close collaboration ever since. And one of the first studies used fMRI to investigate how aging affects neural specificity. So we scanned 13 college-aged young adults and 12 older adults whose average age was 70. And everyone read words, looked at pictures of faces, and looked at pictures of places, while neural activity was estimated with fMRI. Now, our findings in the young adults looked a lot like the previous findings, demonstrating neural specificity. I mean, in virtually every young subject, we found an area that responded selectively to words, and another area that responded selectively to faces, and yet another area that responded selectively to places. In contrast, the neural responses in the older participants were much less selective. The region that responded most to words, it also responded a lot to faces and places. The region that responded most to faces also responded a lot to words and places. And the region that responded most to places also responded a lot to words and faces. In short, the differentiation between neural responses was significantly reduced in the older people. And this phenomenon is now often referred to as age-related neural de-differentiation. So neural representations seem to get more confusable and less distinctive as we age. Does that make any difference? I mean, in particular, do older people who exhibit the most neural distinctiveness perform better than older people who exhibit the least? Well, two of my graduate students, Jun-Koo Park and Joshua Karp, collaborated with me, Dr. Denise Park, and one of Dr. Park's research assistants in order to investigate that question. We scanned 19 participants who were in their 60s and another 19 who were in their 20s. And everyone performed a simple visual matching task on pictures of faces and on pictures of buildings. And we then measured the distinctiveness of the neural activation patterns associated with the faces and the buildings in each individual participant. Now, consistent with our previous study, many of these older participants exhibited neural de-differentiation. That is, the neural activation patterns evoked by faces and by buildings weren't nearly as distinctive as they were in the young adults. On the other hand, some of the older participants looked a lot like the young adults. Their neural activation patterns were still quite distinctive and easy to tell apart. We also asked all of these participants to perform a bunch of cognitive tasks that typically decline with age, such as executive function tasks and processing speed tasks. But we also gave them a task that typically doesn't decline with age, namely a vocabulary task. Now remember, fluid processing like executive function and processing speed, that tends to get worse with age. But crystallized knowledge like vocabulary doesn't. Finally, we analyzed the relationship between neural distinctiveness and cognitive performance. And can you guess what we found? Older people who showed the most neural distinctiveness also performed the best on measures of executive function and processing speed. Conversely, people exhibiting the least neural distinctiveness performed the worst on these tasks. But this relationship was only observed for the tasks that decline with age. For the vocabulary test, how distinctive a person's neural representations were didn't make any difference. So neural distinctiveness affects performance on tasks that tend to decline with age, but it doesn't affect tasks that don't. And that supports the idea that changes in neural distinctiveness may be an important factor underlying age-related cognitive deficits. Okay, let's turn to a different age-related change in brain function that was also discovered using fMRI, namely age-related changes in signal variability. Now, what do we mean by signal variability? Well, fMRI takes a picture of blood oxygen levels every couple of seconds. So the blood oxygen level is the signal that's being measured. And as we discussed, each image of the blood oxygen signal throughout the brain provides a snapshot of which parts of the brain are most active at a given point in time. And by stringing those images together, you get kind of a movie that shows changes in brain activity. Now, suppose you zoom in on 1 cubic millimeter of the brain and you watch blood oxygen levels over time. What you'll find is that the signal will vary, even if the participant is just resting and doing nothing. That's what we mean by signal variability. The blood oxygen signal is variable over time, and that's true everywhere you look in the brain. But why does the blood oxygen signal vary, even when someone is at rest and isn't doing anything? Well, one very natural assumption is that maybe the signal is just noisy. After all, no instrument is perfect, and fMRI is no exception. So you might expect to see variability in the fMRI signal, even if the blood oxygen levels are actually perfectly constant. And for a long time, fMRI researchers assumed that fMRI signal variability was simply noise and that it could be safely ignored. But in 2010, Doug Garrett, Cheryl Grady, and their colleagues at the University of Toronto demonstrated that that assumption was wrong. They measured fMRI signal variability throughout the brain in about 50 people, while they did nothing but stare at a fixation point. And they reported a couple of interesting results. For one thing, signal variability declined with age in a number of brain areas. And these results were fairly consistent across the participants. Most of the older participants showed decreased variability in the same regions relative to the young participants. Now, if signal variability was just noise, then you wouldn't expect that kind of consistency across brain regions and participants. So apparently, fMRI signal variability is actually conveying some important information that's relevant to aging. It isn't just noise. In fact, Dr. Garrett and his colleagues found that they could predict someone's age pretty accurately just by looking at the variability in their fMRI signal. Furthermore, variability-based predictions of age were far more accurate than predictions based on average blood oxygen levels. That is, the variability of neural activity may actually be more relevant to aging than average activity levels are. And in 2011, these same researchers showed that signal variability was strongly associated with behavioral performance. In particular, they found that people who exhibit greater variability in their fMRI signal tend to be faster and more consistent in their performance on speeded tasks. And as we already saw, younger people are the ones who tend to have the most signal variability. So perhaps their increased neural variability contributes to their behavioral speed and consistency. An important open question is why increased variability would help performance. Well, one possibility is that it prevents us from getting stuck in a mental rut. So just like bouncing around when preparing to return a tennis serve might make us more prepared to quickly move to the left or right as needed, likewise, neural variability might improve our mental flexibility. But the truth is, we don't really know for sure yet. Okay, so we've seen that neural distinctiveness declines as we get older, and reduced distinctiveness is associated with worse performance on tests of executive function and processing speed. And likewise, signal variability tends to decline with age, and reduced variability is associated with slower and less consistent behavior. Now let's turn to how age affects communication between different brain areas. One of the classic studies in this area was carried out by Adam Gazzaley, Mark Desposito, and their colleagues at the University of California at Berkeley in 2005. They were looking for a neural mechanism that could explain age-related memory deficits, specifically in the ability to remember a picture after a delay, despite distraction. So they showed a group of people in their 20s and another group of people in their 60s intermixed pictures of faces and outdoor scenes. And they asked them to remember one group of pictures while ignoring the other group. And they also used fMRI to estimate neural activity while people did this. Now, you remember our discussion of neural specificity. We pointed out that different parts of the brain respond selectively to different categories of visual stimulus. In particular, faces tend to activate a different part of visual cortex than outdoor scenes do. And when Gazzaley and company tracked neural activity in the scene selective region, they found something interesting. When young people were trying to remember scenes, neural activity in the scene selective region was enhanced. But when they were trying to ignore scenes, activity in the scene selective region was suppressed. Now, a natural explanation is that the scene selective region was being modulated by another part of the brain. and that this other brain region was exciting the scene-selective region when scenes needed to be remembered, and it was inhibiting that region when scenes needed to be ignored. Any idea what other brain region might exert that kind of executive control? It's the executive of the brain, the prefrontal cortex. And numerous studies have now demonstrated that prefrontal cortex does indeed excite and inhibit other parts of the brain, depending on task demands. At least it does in young adults. But what about in older adults? Well, Ghazali and colleagues found that their older participants exhibited the same kind of enhancement effects as the young people did. So when the older folks were trying to remember scenes, activity in the scene-selective region went up. However, the older adults did not exhibit the normal suppression effect. That is, when they were trying to ignore scenes, activity in the scene-selective region didn't go down. So the older participants had a selective deficit in neural inhibition. Although their prefrontal cortex could turn on relevant parts of the brain when necessary, it didn't do a good job in turning irrelevant brain areas off. Now, a natural question is whether that kind of deficit in neural inhibition could contribute to age-related problems with memory. It certainly seems plausible. After all, if you have a hard time suppressing the processing of irrelevant information, then it seems likely that you'd get distracted and have a harder time focusing on the information that you want to remember. And sure enough, that's exactly what Dr. Ghazali and his collaborators found. Specifically, older people whose neural inhibition was intact did better on the memory task than did older people whose neural inhibition was impaired. So this experiment suggested that the ability to inhibit neural processing might be a crucial component of successful memory. More generally, a number of studies have found that as we get older, we tend to have a harder time turning off brain systems compared with when we were young. For example, consider the so-called default mode network of the brain. It turns out that your brain is active all the time, even when you close your eyes and try to let your mind go blank. And that resting state activity is not random. It actually involves a consistent network of brain regions in all normal adults. And that set of brain regions has come to be called the default mode network, reflecting the hypothesis that activity in this network corresponds to the brain's default state when it's in wakeful rest, but not trying to perform any particular task. However, as soon as you want to perform a specific task, like talking or reading or solving a problem, then the default mode network gets turned off and other areas of the brain become active. At least that's true in young adults. It turns out that another consistent finding in the aging literature is that older adults have a harder time turning off their default mode network compared with young people. They also don't turn it on as much when they're at rest. Even the communication between regions in the default mode network declines with age. The bottom line is that as we get older, we don't regulate this default mode activity as well as we used to. And that kind of dysregulation is associated with behavioral impairments that we associate with age. For example, Jessica Demoiseau and her colleagues at the VU University Medical Center in Amsterdam found that older people whose default mode network was less active at rest performed worse on speeded executive processing tasks, compared with people whose default mode network was more active at rest. Likewise, Jessica Andrews Hanna, Randy Buckner, and their colleagues at Harvard and Washington University found that people who exhibited greater communication between regions in the default mode network performed better on tasks of executive function, memory, and processing speed. Okay, let's summarize where we've gotten so far. We've seen that age-related declines in neural specificity, in neural variability, and in neural inhibition, and in default mode regulation are all associated with age-related behavioral impairments. But I promised you some good news, so let's turn to that. The good news is that there is now a lot of evidence that our brains don't take this kind of age-related decline lying down. Rather, our brains reorganize their processing in an attempt to compensate for these impairments and to maintain our cognitive abilities as long as possible. Now, some of the first evidence for neural reorganization came from Cheryl Grady, James Haxby, and their colleagues at the National Institutes of Health in 1994. They measured neural activity while older and younger people performed a face matching task and a location matching task. And they found that young people activated posterior areas near the back of the brain more than older people did. But the older people activated anterior regions near the front of the brain more than the younger people did. And this kind of posterior to anterior shift in aging has now been replicated in imaging studies of attention, visuospatial processing, working memory, and episodic memory. Furthermore, Simon Davis, Roberto Cabeza, and their colleagues at Duke University reported evidence that this reorganization actually helps cognitive performance. In particular, older adults who exhibited more activation in anterior regions, like the prefrontal cortex, also performed better on an episodic memory test and a visual perception test. Now, one natural interpretation of these results is that older adults compensate for impaired processing in posterior areas by increasing activity in anterior brain regions. Additional evidence for age-related neural compensation comes from studies of the lateralization of brain activity. That is, the extent to which brain activation is restricted to a single hemisphere versus both hemispheres. Roberto Cabeza, my colleague Patricia Reuter-Lorenz at Michigan, and a number of other researchers have repeatedly found that cognitive tasks that evoke activity in a single hemisphere in young adults often evoke activity in both hemispheres in older adults. Scientists would describe this pattern by saying that activation is lateralized in young adults, but bilateral in older adults. For example, retrieving items from episodic memory often elicits activity that's strongly lateralized in one hemisphere in young adults. But the same tasks have been shown to activate both hemispheres in older adults. Likewise, working memory tasks typically produce activity that's strongly lateralized to one hemisphere in young adults. But in older adults, both hemispheres get activated. And that same pattern has now been reported in tasks of perception and inhibition. In fact, the pattern is so common that Dr. Cabeza gave it a name, HERALD, which stands for Hemispheric Asymmetry Reduction in Older Adults. Now, a number of studies have also provided evidence that this HERALD-like neural reorganization actually leads to improved cognitive performance and is therefore compensatory. For example, Dr. Cabeza and his colleagues asked people to perform memory tasks and measured the lateralization of brain activity in young adults, in low-performing older adults, and in high-performing older adults. Now, as expected, the young participants exhibited good performance and strongly lateralized neural activity. Furthermore, low-performing older adults also exhibited a similar lateralized pattern. But the high-performing older adults activated both hemispheres. That is, a reduction in hemispheric asymmetry was only found in the high-performing older adults, suggesting that activating both hemispheres actually helped their performance. And many other scientists have now endorsed this compensation interpretation, although it's still a topic of ongoing debate. Okay, let's wrap up. In this lecture, we examined a number of ways in which brain function changes as we age. Neural representations become less distinctive, neural signal variability declines, and we become less able to inhibit neural processing and to regulate activity in the default mode network. And unfortunately, all of those changes are associated with cognitive impairments. On the other hand, there's growing evidence that our brains also reorganize as we age in ways that may compensate for age-related deficits. In particular, neural activity appears to shift from posterior to anterior regions and to become more bilateral, and both these changes have been associated with improved performance. So, although some of the news is bad, there's also some good news. And later in the course, we'll explore strategies that we can adopt to help our brains compensate so that we can live longer, healthier lives.